Sanger sequencing has been the foundation and gold standard of DNA sequencing since 1977. While new sequencing technologies have emerged since, Sanger is still an invaluable tool for all kinds of labs. The basic steps of Sanger sequencing are simple. The reaction involves template DNA, a primer, DNA polymerase, nucleotides, and fluorescently labeled dideoxynucleotides. First, the DNA is denatured and then cooled to allow the primer to bind. The DNA polymerase enzyme then lengthens the strand according to the DNA template until it randomly places a dideoxynucleotide. The dideoxynucleotide is missing the hydroxyl group, which allows the next nucleotide to bind. So once it is placed, the chain is terminated and will not grow any longer. The color of the fluorescent dye on the dideoxynucleotide represents the last base which was added to that fragment. This reaction is repeated until the sample contains the fragments of all different lengths, each terminated by a fluorescently labeled dideoxynucleotide. When the fragments are separated by size, the dyes can be read in order to give the sequence of the template DNA.
Capillary electrophoresis is a method for separating and identifying fragments of fluorescently labeled DNA by passing them through very thin polymer-filled capillaries. The use of capillaries instead of a conventional slab gel makes it possible to apply a much stronger electric field, resulting in faster separation and higher throughput. The process features single-base resolution and requires small sample quantities. Applications include STR analysis and Sanger sequencing, as well as many other everyday applications used by life science and forensic laboratories. Here's how it works. First, fluorescently labeled DNA samples are prepared for electrophoresis and loaded into the CE instrument. The capillary array is filled with polymer and placed into the sample wells. When the capillary array enters the wells, an electric current is formed and the DNA fragments are moved electrokinetically into the capillaries. Next, the ends of the capillary array are inserted into the cathode and anode buffer. The instrument applies a specific voltage and temperature to control sample migration through the capillary array. The fragments are separated by size, with the smallest fragments moving fastest. The final step is sample detection. As the fluorescently labeled fragments move past the detection window, a laser excites the fluorescent dyes. The emitted light is spectrally separated and recorded by a CCD camera. The CCD camera records the fluorescent signature of each fragment, as well as the elapsed time for the fragment to migrate through the polymer. The raw data is sent to the collection software in the form of a peak, and is now ready for analysis using your choice of software program.
Nanopore sequencing is a third-generation sequencing method used for the sequencing of biomolecules like DNA and RNA. Most of the DNA sequencing methods require the target DNA to be amplified by PCR. However, nanopore sequencing method can sequence single molecule of DNA or RNA without the need of PCR amplification. The nanopore sequencing machine is very small. It uses a small chamber having a hole with few nanometers in diameter. The nanopore can either be solid state or made up of biological molecule. DNA or RNA is introduced in the chamber through the sample inlet. When an electric field is applied, the DNA or RNA starts moving to the other side of the chamber through the pore. During this process, a small voltage is applied across the pore. This voltage in turn gives rise to current, which can easily be measured. Now here comes an interesting thing. The magnitude of the current depends on the base composition of DNA or RNA. Hence, just by measuring the magnitude of electric current across the nanopore, the base composition of the polynucleotide chain can be obtained. The nanopore machine is also very small and portable, which gives a good advantage over other DNA sequencing techniques.
So now let's see about DNA microarrays, which are also known as DNA chips. These are tools which are used for gene expression analysis and for genotyping. These chips may also come up in different flavors. DNA microarray data is useful in computer science for bioinformatics applications like disease prediction, gene expression analysis, and personalized medicine. It enables machine learning models to identify patterns in genetic data, aiding in drug discovery, biomarker identification, and genetic network analysis, which thereby helps us to improve healthcare through computational approaches. The first step for microarray is the sample preparation. In this step, we have two samples. The thing that we see here, we have two samples. Samples 1 might be tissue from a healthy cell, and sample 2 could be cells from tumor tissue. The RNA is extracted from both samples and then transcribed into cDNA using reversed transcription. During this step, the cDNA is fluorescently labeled, and the cDNA from the healthy sample 1 is green and cDNA from the malignant sample is in red. So now sample preparation is complete. So when we take a closer look at the DNA microchip, a microchip has many spots. Looking more closely into one of them, each spot is coated with multiple identical probes. These are short oligonucleotides that cover the sequence of a specific gene. Now we can observe the spot of gene X when we add our samples, in this example for molecules from sample 1 hybridize with the probes of that spot. Sample 2, however, does not contain complementary cDNA molecules and is washed away. The spot we are looking at appears green after laser scanning, which tells us that gene X is expressed in sample 1 but not in sample 2. Examining a different spot of the microarray containing different oligonucleotides against gene Y, we might observe that opposite scenario with only sample 2 containing complementary cDNA but not sample 1. So in this case, the spot of gene Y appears red after scanning because of the specific fluorescent label. As a conclusion, we can say that gene Y is not expressed in sample 1 but in sample 2. The third possible scenario in both samples contain molecules that may hybridize with a gene's probe as in shown here for a hypothetical gene Z. After laser scanning, the spot of gene Z looks yellow because both fluorescently labeled samples are present after washing. This result tells us gene Z is expressed in both samples in the healthy tissue and also in the tumor tissue. In reality, the cDNA sample is added to the whole plate in one step. During microarray chip analysis, a laser excites the fluorescent dye and the emission levels from each of the spots are measured by a detector. This emission level, which is the raw data for computer scientists, can then be analyzed using bioinformatics tool. So, as discussed in class, we know that raw gene expression values. So, what we see over here is what I typically showed you in class. Each row represents a gene and each column represents a sample. These values show how actively a gene is expressed in different conditions. But how do we determine if two genes are related? To measure similarity between genes, we use Pearson correlation. This gives us a score between -1 and none, where 1 means a perfect positive correlation, meaning the genes behave similarly, and -1 they behave in opposite ways, 0, no relationship. Here we see some genes have high correlation values. In this graph, you can see the values in the table. We can see high correlation values, meaning they are highly likely to be co-expressed and may perform similar biological functions. But how do we determine which genes are significantly related? So we apply a threshold of 0.8, meaning only genes with a correlation of zero point 0.8 or higher are considered strongly co-expressed. The result in this binary adjacency matrix, which 1 means a connection exists and 0 means no connection. Now let's visualize these relationships in a graph in a gene co-expression network. So you see the graph here. Each node represents a gene and each edge represents a strong correlation. This helps data analysts or scientists detect clusters or co-expressed genes. You can identify clusters or communities using any of your data mining algorithms that we discussed, which might be involved in similar biological pathways like disease mechanisms or drug response. So on the whole, we start with gene expression data, we calculate Pearson correlation to find similarity, we apply a threshold to filter strong relations and connections, we build a co-expression network to uncover meaningful relationship. So these methods are widely used in bioinformatics for gene function discovery, disease research, and even AI-driven healthcare solutions. Thank you.